Okay, so today we're going to cover the association areas. In the first lecture, we covered how the eye works, and then we covered how the visual system analyzes the eye's information and where the visual information goes to. And now, in one quick lecture, we'll take care of the rest of the brain. So it's a quick lecture on what the rest of the brain does. And so not functionally, but structurally, the brain is made up of these four lobes. The frontal lobe here in the front, and then there's this thing going down the middle here is your central sulcus, and it divides the frontal from the parietal, which is a second area. Then back here, there are a lot of structures here on the visual system, and this is called the occipital. And finally, on the side here, somewhat like a wing on a bird, is the temporal lobe, because it's not quite attached in this front portion here, and it's a big sulcus called the lateral sulcus, where Sylvian fissure comes down here along the side. Now, besides that, there are 5 functional subdivisions, and we saw there's the primary sensory, higher order, the association areas, which we'll largely cover today, and premotor and primary motor. So the primary sensory areas, V1 was one of them. We took how, directly from the eye, you go through the thalamus and then go to V1. Well, similarly, you go through the thalamus and get to other primary areas. And one of them is the somatosensory cortex, which is right here, just behind the central sulcus. Okay, and then over on the side here, within the temporal lobe, is the auditory cortex, and elsewhere you got things like vestibular and taste represented. From there, signal moves to higher order areas, and these higher order areas are beside the primary. So we saw that from V1 we went to V2, V3. Similarly, over here, from the primary sensory, you go to secondary sensory areas, and around the auditory you've got higher order auditory areas. From there, you move on to the association areas, and there are basically 2 of them. One, the prefrontal, taking up most of the frontal lobe, just a little bit here and here left over, and then the parietal temporal occipital, because it takes up the temporal lobe, the parietal lobe, and a little bit of the occipital. And in these areas, these association areas, we find that different modalities combine. So touch and vision combine. So you can touch something and recognize what it is, and see something and recognize what it is. You involve planning, making plans for directing movements. You store things, you remember things, like what's in today's lecture. And in humans, these association areas occupy something like 80% of the cortex. Whereas in lower species, they take up much less. So much of what you do you can consider as a reflex response, and you have to have an output, which is something to do with your motor system, be it able to say what word you're reading, or move a mouse, and it's directed by some sort of sensory input. So basically a reflex. So it could be something simple like swatting a fly, or something more complex like writing down the name of something you see. And these more complex things they require processing throughout these areas, including these association areas. The more simple things, like swatting a fly, all you need is a connection between your primary and your motor areas. Now, we saw before that the brain is largely confined, or the important parts of the brain is largely confined to this gray area that surrounds the cortex. And it's you see a lot of folds here. And that folding is done so you can increase its volume. You can imagine a paper bag. When it's blown up, it's quite big. But when you crumple it, it becomes quite small. So you can fit a paper bag in the tiny little crumple, and all that its covering is confined in a small volume. So the same here. We've got all your neurons, the 100 billion of them, are located in this gray matter. And you want as much of it as possible, because within those synapses, the connections to these cells, is where all your memories are stored. This is where the important part of the brain is. And with the brain is also extensively interconnected. You see all this wiring coming through it. And the wiring occurs, as we mentioned before, through this white matter. These are where all your axons lie. And the axons could be somewhere very short, like V1 connecting to V2, or somewhere a little bit longer, like V1 connecting to MT, or something longer still going to the opposite cortex through the corpus callosum. And this extensive connections are what the important thing about the brain is that it's extensive connections, but it's also what makes the brain susceptible to things like epilepsy. So if something wrong happens here, this area starts firing. This inappropriate activity quickly spreads throughout the brain through these extensive connections. Now, what more remarkable than the 100 billion neurons is that we have 100 trillion connections. Though that's much bigger than the amount of terabytes on any big disk these days. Now, the gray matter requires a lot of oxygen. It requires more oxygen than the white matter. And we're over the last 10 years, we've started to use that fact to image the brain, because we can measure how much oxygen is being used using fMRI. So areas of the brain that are more active have a different appearance when you do a magnetic resonance of them, largely because, as you may remember, the hemoglobin in the oxygen that your red blood cells, your blood cells provide, has a different nature when it's oxygenated versus when the oxygen is removed. And that property is what changes the magnetic resonance properties of that region and tells us how much oxygen is being utilized, and therefore we can assume how much activity there is in that one little part of the brain. Now, and so we can measure the activity of these little parts. You can put people in magnets and test their visual system and see how well, determine whether it's V1, V2, V3 that's being activated. And this area, this technique is now being used clinically to determine what damage has occurred to the brain. Previously, we could just look at the brain and see its structure. We can see whether there's some sort of disease process or tumor in a place. But now we can actually see if brain cells are being active. And one of the sort of star members of the BMI, Brain and Mind Institute, is a fellow by the name of Adrian Owen. And he used this technique to determine if people in comas, so they're in vegetative comas, they've been lying in bed for years and nobody knows if they're conscious or unconscious. And he determined by using this fMRI technique, to find out that they are conscious. They asked these patients questions that would light up certain parts of the brain. If the answer was yes, think about this. If the answer is no, think about something else, so another task. And with this yes-no answer, they found out this person was entirely conscious of what was happening around him. Now, this connectivity, the amount of connections we have, is what distinguishes us from computers. I think it's going to be still a while before we have AI take over, because each of the 100 billion neurons makes 1,000 connections to 1,000 other neurons. Okay, and each with very strong synapses, 10 synapses per neuron. But the important thing is that it connects to 1,000 others. And it's far more than any PC, which has sort of 64 wires running from its processor to different parts of its memory. Okay, to give you an impression, okay, let's suppose you were using Twitter, okay, and everyone in the world was connected to Twitter, okay, and each time you sent a Twitter, it would connect to 1,000 other people on Twitter. You had a lot of followers. How many times, and each one of those could connect to 1,000 others, how many people would it take to contact everyone in the world? How many tweets? Do you think it's 3 or less? 4? 4 to 10? Or more than 10? Okay, we'll do hand raising this time. Okay, who's 3 or less? Okay, who's 4? Okay, who's 4 to 10? Okay, you can't be neutral here. 10 or more. Some people are neutral. So what's the answer? Well, surprisingly, it's 4 tweets. In 4 tweets, you can connect to everyone in the world. More than everyone. You can connect to 1 trillion people if each of these tweets run forward. And that's what makes the brain so powerful. So in 4, one signal goes to 1 neuron, that goes to 1,000 neurons, and then turn to 1,000 times 1,000, and so forth. And in 3 connections, you've talked to every neuron there is. You've sent the signal everywhere. So it's going to take a while for computers to catch up. And here you see a nice diagram of all these connections in the brain. And they're using again, a form of MRI called diffusion tensor imaging. And so they can image the connections from one part of the brain to another part of the brain. They can determine if, what areas connect your fusiform face area to other areas. And they're using that to come up, they did a study, spent about 5 years making the human genome. Now they're doing the same thing to make the human connectome, getting all the connections of a human brain mapped. That's a big project, and if you want to take a look at some of it, do so from this connection here. Okay, we back up to where we were in the second lecture. The cortex has these 6 layers. Again, the topmost surface is layer 1, the bottommost next to the white matter is layer 6. And again, the input comes into layer 4, and the output comes out of layers 3 and 5. And the thickness of the layers tells us a lot about its function. So, for example, we remember that layer 4 was very thick in the primary visual cortex because it was receiving such a large input. Layers 3 and 5 are very thick in your motor cortex, so the areas of the brain that produce the output. And a fellow by the name of Brodmann, about 100 years ago, he looked at the brain and saw these changes in thickness from area to area. So the thickness from V1 to V2 occurs when this area 4 suddenly reduces in thickness. So Brodmann could tell this was a different area. And he went along and divided the brain up into 43 different functional areas. And we're using fMRI and modern techniques, we're rediscovering that in fact these are 43 different areas. But not only that, we're finding that within each one of these areas, there are many subdivisions. And all these areas have columns. So what do all these areas do? Well, there's two theories. One is called Lashley, and he thought that one part of the brain was largely indistinguishable from another part of the brain. So that if you lost a few neurons from a small lesion, that was not important because it would have a minimal impairment. So but the evidence against that is the brain isn't uniform. The eye projects to V1, not to someplace else. Your motor cortex that produces all the signals down your spinal cord is located in front of the central sulcus and no place else. So there is differences in function. The other extreme theory is that there's something called a grandmother cell theory. So you go from simple cells to complex cells to end-stop cells to cells that recognize faces, and finally to a cell that recognizes grandmother. Okay. Well, it's partially true. There are a group of cells, and it probably turns out to be millions of cells located in the fusiform face area that light up for your grandmother and nobody else. Okay. But it isn't just one neuron, and so when that one neuron dies, when one neuron happens to die, you don't suddenly wake up in the morning and can't remember your grandmother. You've got some resistance to that. And in fact, most of you, after about early childhood, are starting to lose the number of cells you have in your cortex. So it's downhill. It's been downhill from early childhood for most of you. Although, as we'll learn later on, there is evidence that some of these cells are being replenished by new cells being formed in your cortex. So probably truth lies somewhere between these two extremes. That is, it's not a single cell, but it's a group of cells, and one group of cells stores grandma's face, another group of cells stores her voice. So she's spread out all over, as this theory would suggest, but not quite as equipotential as Lashley thought it was. So is the particular area different from person to person? No, because it's very plastic early in life, and the particular part is missing or has some defects. Another part will take over. So if you had, we saw that if you had a defect in one eye, the other eye would take over. But similarly, each one of us has slightly different combinations of sensory modalities that one's exposed to early in life. And so as a consequence, you'll develop slightly different connections. And then if a particular area is damaged, that is, it loses its function, another area will take over. So language, for example, is usually represented in the left. But if you have damage in early childhood, that area will move to the other side and be represented just as functionally on the right side. Now, and also the two halves of the brain aren't identical. One side, and that's usually called the dominant side, it's usually the left, has certain functions, and the other is called the non-dominant side. Now, it's given the word dominant, yes?
I had a question about the last thing you said about the language.
Yes.
Cortex. So if it expands into the right hemisphere, will it take away from another cortex's functions, or will it be implication?
It will no doubt diminish something because you only have a finite number of neurons. But given that you still have 100 billion and 100 trillion connections, that difference, that we haven't measured what it is that you've lost as a consequence. So it's not measurable or hasn't been measured yet.
Do you think it would kind of prioritize the function of what would be more important to keep?
Can you prioritize what you keep? I don't think we've gotten to that stage. Yeah, I don't think we can. I don't think there's much chance you could do that. So, in terms of dominant and non-dominant, the left side is usually called dominant because that's where the language lies, and in most people. And when neurosurgeons started poking into the brain, they discovered that this side was involved in language, and therefore they tried to stay away from it, because if you lose language, it's obvious. But if you lose some part of the right cortex, it's less obvious. And so they gave the left the name dominant because they thought language was dominant, and the other side a lesser non-dominant name. But that's not, that name, dominant, non-dominant, is I think, misleading a lot of people. I think the right side is as important, if not more important, than the left. The left side is usually involved in sequential or serial tasks. So, for example, language is a sequential serial task. When you make up a sentence, you make up a series of words. Your brain has to construct this series of words. And when you read a sentence, you read the whole sentence, and then you know its meaning. So you decode the series of words. And it's also good in things like math, particularly algebra, where you're doing a series of tasks to discover what whether A equals B or not. The non-dominant side, usually the right, is more involved in parallel tasks. So one good example of a parallel task is your ability to recognize a face. When you look at the face, it's all the features of the face that suddenly are put together to enable you to recognize this face. And the fusiform face area is larger on the right side of most people than it is on the left. It's also good in things like geometry, when one looks at triangles and seeing, well, this triangle is similar to that triangle because it has the same angles, in spite of the fact that one is smaller and the other is larger, or when it's turned some way. And you can, that an I represents an L, resembles an L, and a C represents an O. And that's also seems to be very important for music, or at least parts of music. Now, one of the things that's interesting is what happens when these two areas, the two hemispheres, become separated. And this was not infrequent. Occurred to calm down some forms of epilepsy. Now you might think cutting the two halves of brains in half is kind of a drastic cure for epilepsy, but for some people that was the only choice. And if you're an epileptic and you have seizures daily, and those are dangerous to your life. Often there are drugs that will quiet down the seizures, and often people can become seizure free with drugs, but sometimes that's not possible. And so before these drugs got good, one of the ways of dealing with seizures is to cut the corpus callosum. And it's interesting to look at what happens to these patients. And what you can, for example, show them an apple, and you have the patient look forward, and the apple appears here on the left visual field, and so it goes to the right cortex. So the right cortex sees an apple, and this arm is the arm area here of the left arm is located here in this right cortex, and so it can reach for this apple. But if you ask this subject what it was he was trying to reach for before he got to it, he could feel it, he wouldn't know that it was an apple. He couldn't say apple, because his language areas were over here on the other side, the side that didn't see the image of the apple. And in studying these patients, you discovered sort of an interesting thing. These patients had these two minds existing in one head. One mind would be thinking of one thing, and the other mind would be thinking of something else. And one subject that they talked about is one patient who, when one side of his head would be very fond of his wife, and the other side of his head would not. And so one side would push the other, the wife away, and the other side would hug her. It's amazing to think of living in a head with two different minds. So what are the advantages and disadvantages of this specialization of each cortex? One advantage is the pathways that go through the corpus callosum are long. So if you're going to do some function, make that function within that same hemisphere, so the connections can be shorter. And duplication is a waste of one's hemisphere. You can specialize in more tasks if you can divide the capacity amongst two. The disadvantage, of course, is there's less redundancy, and redundancy is good if some part breaks down. Okay, let's now look at the function of the association cortices, these two different ones in more detail. The first one is the prefrontal association area, this area up here in front. And this has especially become larger in Homo sapiens. They call us sapiens because of our wisdom. Sometimes one doubts that we are the wise species in this world, but that's what we are named. And you can see here how the percentage of the brain has the frontal lobe as the percentage of the total brain has increased in humans. Now, why do you think this happened? Why has Homo sapiens developed such large brains, in particular large frontal cortex? And the answer is surprising. It's cooking. Our ability to cook food is what allowed us to develop such large cortices. Because again, this small part of your total body, in terms of body weight, it's only 2% of your whole body weight. But it uses up 20% of its oxygen and 20% of all its calories. And so if you weren't able to cook food, and that is to digest the contents of food quickly, that's what cooking does. If you have to eat wheat, it'll take you a long time to digest raw wheat than it would after it's cooked. And so cooking is what allowed us to develop such large cortices, because without it we'd have to be eating and finding food all day long. Okay, let's test your prefrontal cortex. Okay, you see this egg here? It'll disappear shortly, and I want you to remember where the egg is. Okay, starting now. Okay, can you remember where the egg is? Is it here? Is it here? Raise your hands for those that think it's here. Good. You've all got good prefrontal cortexes. This is a test that seems simple, but it uses a lot of things. First of all, it's spatial, so you're remembering something in space, and secondly, it's delayed. So you have to hold this location in your mind over time, and so this is an important thing that the prefrontal cortex does. And young children before the age of 1 don't have this area well developed yet. And so if you hide a toy underneath the cover, the child will forget that there's a toy there. This is another one of those noisy examples. Okay. What was the difference here? Well, we'll play this noisy example over again. There's a V1 cell, and there's a prefrontal cortex cell. So you could see something lit appeared here, and this prefrontal cortex cell lit up in V1 because that image appeared there. But then it went off, and this V1 cell went off. But this cell here in the prefrontal cortex remained active. And it remained active largely because of something called working memory. It's a form of memory, but it's a short-term form of memory. And this is a very important ability of the prefrontal cortex, to hold things for a short period of time. But short is a little bit undefined. And you use it for things like playing chess, for example. To play chess, you've got to plan moves ahead of time. You've got to imagine the chessboard. If I move this, and then what happens if I move this, and I move this, and you've got to hold all those sequences in your prefrontal cortex. And so this is what us humans excel in, being able to hold so many things and think ahead in terms of steps, imagine things. Now, this area is very dependent on dopamine, and dopamine then, not having any dopamine, impairs working memory. And people with Parkinsonism have trouble remembering things in their short-term memory. And many people with old age, Alzheimer's, also have the same trouble. And so with Parkinson patients, for example, they have trouble initiating movements, and they need triggers. I don't know if you've seen this movie, I think it's called Awakenings, and it shows people before they discovered that they could give dopamine to these Parkinson's patients. What they did was have these patients were largely catatonic. You would stand them up, and they would stand there. You would sit them down, they would sit. So this doctor, he lined up these patients, a row of 8 on one side, a row of 8 on the other side. They faced each other, and then he threw a basketball in between the two rows. And these patients who had been catatonic suddenly bounced this basketball back and forth between these two rows of patients. Because they had this visual stimulus there, they didn't have to imagine where they were going to. They just went for it. So they're not dependent on their working memory to produce these movements. They saw them happening. So you could, again, if you can imagine the chess moves, you can make plans, you can plan what your chess game is going to be. But also, if you don't have this area, you become calmer and less frustrated. So if you weren't making plans for a chess move, then if things went wrong with this game, you wouldn't get frustrated because you'd never made that plan to make that move. And so, because people became calmer after lesions, one popular cure for people with acted very frustrated and angered at not making the right decisions was frontal lobotomies. It is amazing how popular this cure became. A fellow by the name of Muniz won the Nobel Prize for coming up with this technique. Okay, he won the Nobel Prize. And this doctor in the States, Walter Friedman, this is a picture of him, and he using an ice pick that he drives with a mallet through the top of the eye into the frontal lobe, and swishes this ice pick back and forth, cutting off the frontal lobe from the rest of the cortex. and lobotomizing this poor patient that's lying here. And around here you can see all the residents taking careful notes of what's happening and not being horrified at what's happening. So I point this out because there's a lot of things that if you go to medicine that you'll be taught that you think, oh, are quite normal at the time. And then 20 years later, you'll look at it like you're looking at this picture and say, oh, that was horrible. And right now, we can't imagine, like they couldn't imagine how horrible it was. Anyways, let's move on to the next area, the parietal temporal occipital cortex. It's a big area, comprises 3 lobes, and it's involved in many important things. One is this polymodal convergence. So it's located here between your visual areas, somatosensory areas, and your auditory areas. And so combine the signals coming from these 3 areas, and you can be able to see words, hear words, or feel words, and react to them the same way. It's also involved with attention, and we'll see in a moment how important attention is. And finally, it's involved in memory, and we'll do more of memory in the last lecture of this course. So attention. I'll show you in a moment how important attention is, but there was a study done with this gentleman here, dressed in a clown suit, riding something like the square in front of a UCC, and the students asked the people around whether they saw this clown. And if you were walking with a cell phone, the odds are you wouldn't notice him. Okay, because your attention was elsewhere. So inattention is like blindness. If you're texting and driving a car, you're as impaired as you would be if you were drunk. And I'll show you, hopefully to convince you that if you don't have attention, you won't remember this or notice this or anything else in this lecture. So, what's changing in this picture? Anyone notice what's changing in this picture? Raise their hands. No one. 1, 2, 3, 4, 5, 6, 7, 8, 9. Takes you a while, eh? Some people still not. Look here. Okay. It's amazing. So once I point it out to you and your attention's focused there, everyone can see that that's what's happening. But unless you have attention focused, you won't see that as happening. And you can think of attention there as the little spotlight that lets information flow from your primary areas, that's where, like if you won, to your association areas. And if you don't have that flashlight focused, that signal won't get to your association areas, and you'll be blind to it. Now, we talked about how the brain tries to minimize the amount of oxygen that it consumes. And this is one of its techniques. So by attending to things, different aspects, you activate not the whole brain, but just a small part of the brain that's involved in that task. So your need to attend to things is largely driven by the fact that you need to pump oxygen into that little part of the brain, and you need to activate those neurons and not other neurons, because if you didn't, if you activated all of them, you'd run out of oxygen. So you can see here something entering your V1, then going through gradually to your association areas and the prefrontal one, where working memory lies. Okay, and it takes time. And then you can see here that if while you're attending to this thing, something happens over here, that signal doesn't get through. Now, sometimes an object will stand out from its background. You remember how a horizontal line stands out from a bunch of vertical lines? Well, you can see if that thing stands out, that then draws your attention to it, and then it's processed and enters your working memory. So that standing out is important. If something flashes, it's also important. You notice it. Now, if you're attending to one thing, you won't be attending to these others. You have to attend to them in sequence. And your brain then can draw its attention to each one of them. So you could use your cortex. Now, before you couldn't see this bicycle, but now this flashes on. So the flashing is what draws attention to it, and that object now enters your association areas. Takes time sometimes to find things that one's looking for. So it takes time to find Waldo. Waldo, I think, is located here. But here we have, Waldo itself doesn't stand out from the background, okay? So it takes a while for you to hunt around and find it. And this taking time occurs because of several reasons. First of all, Aldo doesn't automatically draw your attention to him, because he's composed of similar colors and orientations as a lot of other things in this thing. And because it takes time for your attention to find Waldo. So you find Waldo either by shifting your attention without moving your eyes, and that's called a covert change in attention, or by actually moving your eyes, making a scan from one spot in the visual field to another spot. When you're looking around the room, your eyes shift from person to person until you find the person that you're looking for. And those are called overt changes in attention. And both these processes take time to move to either making overt or covert movements take time. And it takes time to decide. Attention isn't the same thing as arousal. Arousal is something that happens when we drink, for example, coffee. And it involves neurons in your underneath your brain, in your midbrain, like the locus coeruleus, sending out like a volume control to every part of the brain, making it more active. But it's sort of like a general change in octane to the brain. It doesn't focus the brain in any one particular area. Okay, I'll show you here what will appear, you'll see an X. You have to fixate it. So try to do that. And then there'll be a yellow circle, and you're supposed to notice it. You're supposed to respond as quickly as possible when you see the yellow circle. But to guide you as to where the yellow circle might be, you'll see a little square. So that'll hopefully draw your attention to where the yellow circle will appear. Okay, I'll start now. Okay. Too late. Okay, I'll do it again. Okay, if you could what could you do? Could you hit the table when you see the yellow circle? Okay. Okay. That was a little slower because it misled you. It was a little slower again because, again, it misled you. It appeared way up here. So you can see how attention is important in noticing things. Oops, went the wrong way. Okay, another experiment. You'll see objects appear here. Hit the table when you see two successive objects. So you see one and then another one of the same color. Good. So here, when you did that task, a part of the brain that's involved in color was being activated. Okay. Oops. Okay, now we'll show you the same thing, but something that's the same shape. Okay, again, a different part of your brain was activated for that. And finally here, when two things are the same direction of motion. Okay, again, so MT was activated then. You can see then how shifting your attention to different tasks shifts the brain activity from spot to spot and drives the energy requirements of that spot. And your brain can operate with the minimum amount of energy consumption. Another test. Can you see the line that stands out? You should see that easily. Okay, try this one. Raise your hand if you see it. Ah, okay, that wasn't too hard. Can I see it? Ooh, forget what color it was. There. Okay. Next one. Well, in this one, okay, let's suppose you knew what color was in advance. So if you knew what color it was in advance, I haven't told you what color it was. Okay. it's going to be black. Okay, see how quickly you can find the black line that's different. So focus on the black lines. Notice how it pops out. Okay, you're looking for black. This line pops out at you. So you can direct your brain to focus in on the color and activate the color areas, and it stands out. So we've got two types of attention. Spatial attention. You can direct your attention at one visual field or the other visual field, or if we focus on different features, color, shape, particular attribute. So we've got attention that can also focus in on objects. So here we can see this hippopotamus stand out. Here we can see this line stand out. And here you can see, if you were concentrating numbers, this A would stand out. Now, the opposite of attention is neglect. If you lack attention, you lack awareness of things. So people with right-sided parietal lesions neglect objects on their left, so on the left visual field. And it's the left visual field of the object. So in this case, the object is this face. So the cortex draws a frame, you can think of, around the object, and it's whatever is on the left of this frame is not visible. And so I'll show you later, after this class is over, how if you have a lesion of the left, of the right parietal, you can be making a portrait of yourself in the mirror and just draw half your face. You would think that, someone who's an artist could look at the scene and know that a face has two sides. But you'll see this artist drew just one half of himself because he was unaware of this other half because of this lesion. Okay, so neglect is being totally unaware of what's there. So it's different from V1. Here we can see we're looking at the X, and here the face disappears if you have a right V1 lesion when the face is on the left, the opposite visual field. But here the face stays visible, at least the right half of it, independent of where it is on the left or the right, but always the left side of the face is invisible. Now, one would think that the opposite lesion, the lesion of the left parietal, would do the same thing, but it doesn't. And why is that? Well, one reason, perhaps, we don't quite know, is the parietal doesn't have the same representations on each side. On the right, you have both the right and the left visual field being represented, whereas on the left, you just have the right visual field represented. And so if you have a lesion over here, then both the left and the right will still be active. You'll still be able to see something over here and over here, and you won't have neglect. But if you have a lesion over here, then this side can't see over here, just over here. Last thing I want to cover is coordinate frames. How do you represent objects? Well, one is in terms of its allocentric position. And the allocentric position is with respect to some frame. Over here we have this apple, and it's on a table. And that the object's position on this table, you could think of it as as being a certain distance this way and a certain distance this way. It's in an object-centered coordinate. In contrast, another way of representing the things is relative to you, not an object, where they are relative to you. And here is me, and this apple is so far this way and this way with respect to me. And me can be a variety of things. It could be your head, your eye, or your body. Now, the best way of thinking about these things is you draw a transparency with me and an X for where me is, or a transparency with the location of the objects. And you can move them around and see how each one changes with respect to each other. So for allocentric, you can see when you move around, these objects relative to you change positions, but with respect to the table, these objects are staying the same. So in their coordinate frame, the allocentric frame, these objects are the same location with respect to the table. And then when the objects move around, the other frame moves around, you can think of you moving with respect to the table. Their positions relative to you move around, but of course, their positions with respect to the table stay the same. And you could have nested frames. You could have the object on a table and the table in a room. What's interesting about all this is that we saw these frames with the streams. We had a where stream heading up over the cortex, and we have another stream heading down below the cortex. And this stream here works in allocentric coordinates, whereas this stream here works in egocentric coordinates. And this, if you think about it, makes sense. So if you're thinking of something about walking to a restaurant, you want to know where the restaurant is with respect to you. Okay? So you want to know whether it's to the left of you, to the right of you, in front of you. Whereas if you want to recognize a face, where the features are with respect to the face is the important cue. Okay, that's it for today.